Good morning, everyone. I'm Jiao Sima, a graduate student from David Gilbert's lab. Today, I will be talking about cis elements that control DNA replication timing and also genome compartment. So this is just a picture I took a few days ago in Harvard Yard, and I thought these guys look like pretty much like Tad. And I actually want to make a point that DNA replication timing are more linked to the compartment, which are long-range interactions among the TADs, rather than individual TADs, which are relatively fuzzy concepts at this moment for exactly how they look like, where the boundaries are, and what you should call it, TADs or loop domains. Okay, so we are replication labs, and DNA replication follows a very specific temporal order through S-phase. A cluster of origins fire temporally synchronously at different phases during S-phase to generate this replication timing profile. And a typical example is shown here. You can immediately identify segments that replicates early or late along the chromosome. And this temporal order of replication is highly coupled with the spatial distribution of chromatin inside the nucleus. So Hi-C divide the nucleus into two self-interacting compartments called A and B, which corresponds to early and late replicating chromatin that are distributed either at the interior of the nucleus or at the periphery of the nucleus associated with nuclear, in close proximity with the nuclear lamina, not associated with. At a smaller scale, local chromatins can fold into TADs, which corresponds to segments that change replication timing during development, which we call replication domains. And I want to emphasize that these TADs are independently regulated than compartments. You can remove CTCF and cohesing and lose all these TAD structures while where compartments remains. Both replication timing and compartments are highly correlated with other functional features of the genome, such as chromatin composition, gene transcription, and mutation signatures. And the relationship among these highly correlated events of the genome are central questions in the field. However, we know very little about the mechanisms that drives compartmentalization and replication timing. So to that end, I want to use CRISPR-mediated deletions and inversions to identify cis elements that are responsible for these processes. So this is our favorite domain. It is called DPPA24 domain, localized on chromosome 16 in mouse embryonic stem cells. It replicates uniquely early in embryonic stem cells and switches to late replication during neuroprecursor cell differentiation. It corresponds nicely with these TADs, which are located in the compartment A, and they are not far away from the nuclear lamina. It has three active genes, DPPA4, 2, MORC1, and MORC1 has an alternative transcription start site. There are a couple of CTCF and cohesing binding sites in the domain with two convergent CTCF binding sites demarcating the boundary. This is the short nascent strand mapped origins, shows where the replication origins, and there are a couple of P300 sites suggesting a potential regulatory element for gene transcription. So the first thing we went after is the boundary. Okay? And we generate deletion that ranging from 2 kb all the way to 335 kb, targeting this CTCF binding site on the left, the boundary. And you absolutely see no change in replication timing, and this is just a passive shift of replication from the DPPA domain. And we also generate this 45 kb inversion, which invert the CTCF binding site orientation, has no effect on replication timing. For the right side boundary as well, and there's no effect. So consistent with the lack of requirement of boundary, CTCF-associated boundary in replication timing at DPPA24 domain, it found that the genome-wide replication timing and compartment are robust to CTCF depletion. So these are the same samples from Alfesh Nora, in which they show use auxin-induced degradation of the CTCF at the protein level and shows that TAs are almost eliminated, but compartment remains. So both replication timing and compartment are resistant to CTCF depletion. So further confirming that the replication timing are mechanistically linked with compartments rather than individual TAs. So you can change TAs, you can change replication timing in the units of TAs during development, but they are not necessary for replication timing. So because these are clearly negative results showing that CTCF-associated bonding and CTCF depletion does not have effect on replication timing, we wonder what in the domain could contribute to that. So to predict where to lead, we generated a very high resolution capture high-C heat map to see the final scale structure of the domain. And we used perhaps fit high-C to cause significant interactions of this region. And if you zoom in to the domain, you can immediately appreciate about 3 sites that looks like mediate the strongest interactions within the domain, other than this cohesion mediated loop and CTCF stuff. And what's more interesting is that they seem to correspond to regions where they have a lot of active chromatin marks and chromatin features. So the obvious next step is to delete all these 3 sites and see what happened to replication timing. So here you go, the result. The black is wild type, and the red is the triple deletion, deleting all these 3 sites. The triple deletion now completely switched to late replication in a similar fashion as in ESD to MPC differentiation, and we can reproduce this result in a different clone. So we think we have depleted all the cis elements that are required for early replication, and we call them early replication control elements, ERCEs. And I'm not pointing to anything particular here. I'm just want to say that I hope you appreciate how dramatic the phenotype is, because I would generate many, many different mutations before this, and which all of them yield very little effect other than the triple deletion. So because it evolves in 3 sites, I generated individual and pairwise deletion to see what's the role of each individual one. So the individual deletion demonstrates a certain level of redundancy, because AB deletion does not seem to have any effect on the domain replication. And also, the pairwise deletion suggests there's some sort of interdependency among the sites, because if you delete 2 of those ERCs, remaining just 1 in the domain, you see the locally earliest replicating point in the domain. However, they fail to reproduce the full earliness, suggests that you need at least 2 ERCs to make a full early replicating domain. And to test the sufficiency of these segments, we also generated a large inversion, 680 kb inversion, in which the segments within this blue square are inverted. And half of it are this TTR, timing transition regions in compartment B, and half of it are, half of the segments is the AB containing DPPA segment. So if you would just invert the sequences, you can see that the DPPA domain now is immediately adjacent to the B compartment. And this is how it looks like for wild type if you just invert the original replication timing. And this is how it looks like for after inversion, on the actual inversion. You can see that the DPPA domain still maintains full early replication after inversion without its original right boundary. Suggests that this AB containing segments are sufficient to maintain early replication. And we found that ERC is also responsible for associate of component A for the domain. So we did 4C using a bait inside the DPPA domain, and you can see that the ABC deleted allele increased interaction with late replicating regions and compartment B as compared to the wild type allele, which to some sort of extent similar as ESE to MPC difference. And this is just a quantification to show that the switch is sort of, to some degree, to the same degree as ESE to MPC differentiation. And I don't have the time to show the data, but we want to point out that this ERC-mediated interactions are CTCF independent. We still see them as the strongest interactions in the alpha CTCF depletion data. And within the domain, these interactions are not restricted within native neighborhood, and outside the domain, they interact with potential ERC elements along the chromosome. So we think that this inter and intra-domain interactions mediated by ERCs are potentially driving the AB compartmentalization of the chromosome. And what about transcription? So in the ABC deletion, we found that the majority of the transcription are gone, suggests that ABC are responsible for transcription activation in this domain. While for some of the genes are expected because we did delete the transcription start site of these genes. However, the DPPA4 gene is still intact in the domain, and its transcription is also lost. And you may wonder, are ERCs just the promoters and enhancers? And the answer is likely no, because I want to point out that this in the DPPA4 domain, this here DPPA4 gene promoter region here, the 4KB deletion, in addition to BC, looks like similar to BC deletion, whereas where ABC deletion, you lose all the early replication. So I'd like to finish up with this co-regulation model. There are a lot of confusions in the field reporting controversial results regarding the relationship between one another. So in some literatures, replication timing contribute to the transcription potency, competency, and in others they say transcription causes early replication. And in other cases, these two events can be uncoupled. And so to reconcile all those and along with our findings, we favor a co-regulation model instead of a cause-effect relationship among the two. And we want to say that we have identified cis elements that responsible for early replication of the domain and contribute to the compartmentalization. They are strongest interactions with the domain and associated with a profile of active chromatins, and perhaps they contribute to the transcription because they are in close proximity, they contribute to the transcription activation by mediating those genes into shared transcription factories. And without those elements at the genetic level, you remove all these activities. So I'm running out of time, but I really want to thank a lot of people that are involved in this project, and I really appreciate all this effort, and I really enjoyed working with them. So I'll be like to take questions.
We got a minute for questions. Any questions?
So do these regions exactly match with origins of replication if you were to analyze them in a fiber assay?
No. So in the first slides I showed where the short nascent strand mapping of the origins, they do. One site B has a small peak, but you see a lot of other peaks outside ABC. So they do not correspond to short nascent strand mapped origins. Yes?
This is beautiful. Do you think these ERCEs, it looks like they are also transcriptional enhancers. Do you think you can decouple their enhancer functions from a role in the replication timing, or is the replication timing truly following the epigenetic marks up by transcription.
Yes, they do have enhancer activities, we agree. But I think at this stage, I think we cannot tell exactly which one is caused which one, and we believe like co-regulation is a better explanation, reconcile all findings and also the confusing literatures.
All right, great. Same question?
Yeah.
And I could tolerate one more question. No more, let's thank Joe again.
Thank you.